Hello, good morning, everyone. I hope you had a good restful week last week that you didn't have school and you had engagement week and that you are refreshed for the second half of the course. So right now you have finished it 50% of the course already, which is great. We still have 50% to go. So there is just the same format for the second half, right? 10% for the quiz number 3, 10% for quiz number 4. There's going to be a 10% lab, and then there's the 20% final test, which is test number 2. And test number 2 is only going to be like the second half of the course, right? So everything you've learned in the first half, lecture 1 and 5, and then the Beetroot lab, we're not going to get tested on that anymore. All right, so today we're going to start on lecture 6. Hopefully you have the notes. It's Halloween, so I asked my kids to pick me like an avatar, and then they decided that I should be a unicorn. Okay, so in case you're wondering why I have that in my video. No need to be so serious all the time. Sometimes it's better when you're in a lighter mood, right, to to learn new things. Okay, so second half of the course we have the cellular reproduction. That's the topic for lecture 6, which is gonna look into the exact mechanism of how we duplicate our DNA and how we split the cell into two. Okay, and after that, we will look at the mechanism of transcription, translation, and what happens if something goes wrong in the process? Like, what if you made a mistake, right? When you copy your DNA, that's going to create like a mutation, and uh, like what are the consequences of the mutation, right? Well, it depends on where it happens and how bad it is. So we're gonna assess all that, and in lecture 9, we'll talk about how we can change the genetic material of an organism, right, including that of humans, using biotechnology, which would allow us to find a treatment for some of the diseases that has a genetic basis to it. And finally, the last lecture 10, it's going to be a nice transition to the second semester where you will be learning the anatomy and the physiology. All right, so that's the overview of what's coming. Let's start with the cellular reproduction slide deck here. We're gonna be doing a lot of notes copying and stuff today, so make sure you have your um, pencil and paper with you. Something's not quite right with my presentation here. I gotta choose primary monitor. Let's see if that works now. Okay, there we go. So in the 1950s, scientists already kind of know that there is some hereditary material inside the nucleus. All right. But they don't know whether it was DNA or if it was protein. I mean, they certainly know at that point that there are chromosome in the nucleus, but you might remember the chromosome, the chromatin, right, in the form of chromatin, is actually made up of both protein and DNA, right? It's DNA wrapped around the proteins, right, for packaging. So they don't know which of these molecules is actually responsible for heredity, right? Like which one actually contains the instruction, right, for life, which one is actually passed on to the next generation. So if you would, there are like two camps of scientists here, right? One group thing is protein, the other group thinks it is a DNA. Okay, so until 1952, there are two scientists, Hershey and Chase. They together conducted an experiment. Okay, and the experiment definitively showed that it is the DNA that is responsible for containing the instruction to life, right? That's the material that gets passed on to the next generation. And so we're going to take a look at their experiment. Let's see. There we go. So our first note for today is the Hershey and Chase experiment. Hershey and Chase experiment. So the purpose of the experiment is to determine whether DNA or protein is the hereditary material. Okay, that's the goal of their experiment. Now, to do this, they actually used something called a bacteriophage. Okay, so they look like this. They actually do. Okay, this is a bacteriophage. Bacteriophage is a virus that infects bacteria. Okay, it's easy to grow, it's easy to maintain, and it's not harmful to humans. So that's the bacteriophage. Now the bacteriophage has two parts to it. There is a protein shell, and then there is some genetic. I mean, they didn't know back then, but there is some DNA in the center. They didn't know it was a genetic material. That's what I mean. Okay, but they know there is a protein coating, and then there's some DNA on the inside. All right. So I have a question for you guys. Give me one element that is found in protein but not DNA. One element that is found in protein but not DNA. Anybody? Well, both protein and DNA has hydrogen though, right? Madison, if you think about it, they both have carbon, oxygen, hydrogen, for example. But what's one thing that's only found in protein and not DNA? Traces of this thing only. Maybe the other way around is easier. Something that's in DNA and not protein, you should be able to answer that. Phosphorus. Okay, so that's right, Kiana. Phosphorus is unique in DNA and not in protein. And for protein, it is sulfur. You might remember there's trace of sulfur, right, in protein. So what happens is they have two kinds of bacteriophage. Okay, two groups of bacteriophage. Right. We have bacteriophage that are grown in radioactive sulfur. Right? So this is sulfur, we can say grown in radioactive sulfur. Okay, and it's the sulfur 35. Which means the protein code is radioactive. is labeled, you can say. Okay? The DNA is not though. Okay? So this is group number 1, radioactive sulfur. That means you can track the location of the protein. That's all it means. Group number 2 here is grown in radioactive phosphate. Okay, radioactive phosphate. So the DNA is labeled. All right, so think of it as like, glowing signals. Only the protein glows here, and only the DNA glow here. Okay. So how does this bacteriophage work? Right. This is a bacterium. Okay. This is the bacteriophage. So the
sorry, on the group 1, does that say protein coat or code?
protein coat. Like the coat.
COAT?
Yeah, like
Coat?
the
Okay.
coat. Yeah, the
Thank you.
coating, the outside, right? So the virus, the bacteriophage, will land on the bacterium, and just listen to what I have to say here. I'm not going to draw it out because you understand why. Okay, so like they inject something, the virus injects something into the bacterium. Okay, we now know that is the DNA that's being injected, right? Because that's the hereditary material, but they didn't know. They just know something went inside the bacterium, and then that something is going to turn into many viruses. Okay, so something gets injected into the bacteria, which in turns make many copies of the virus. That's how all viruses work, okay? Most viruses, okay? They inject something into the bacterium, and then the that something is the instruction to make more virus, right? So they want to find out what that something is, okay? Now, the um the way that this works is this. Once they let the virus infect the bacterium, and then you do it in like a test tube, okay, basically, what happens is they can take a test tube that has the virus and the bacteria, they let them infect the bacteria, and afterwards, right, they would put this in a centrifuge. Put the mixture in a centrifuge. Okay. Centrifuge is this machine that spins at really, really, really high speed. We use it all the time in many applications. For example, if they want to test your blood, they can put the blood in a centrifuge to separate the contents. So after the infection, they put the mixture in a centrifuge, and then they basically spin it down. They separate it. Okay, so now we have, this is a test tube, by the way, if you don't know what I'm drawing. Right, this is a test tube. And then it will separate into two layers. Okay, the bottom layer is going to be the heavier stuff. What do you think? Do you think the virus is heavier or the bacteria? Which one is bigger anyways, like in real life? It's the bacteria bigger or the virus bigger? Okay, according to my drawing, yep, okay, bacteria is bigger, right? Okay, so the bacteria is going to go to the bottom. So this bottom section is going to be the bacteria, plus whatever was injected into them. Okay, and then the top is just part of the virus that didn't go in. Okay, I'm just gonna pause for a second to make sure you understand the setup. So you let the virus infect the bacterium, you spin it really fast to separate. Okay, so whatever didn't go in stays on top. And then the bacteria, which is heavier, and with the stuff that was injected into it, will sink to the bottom. So any question before we move on? Do you have a concept question? What's a concept question?
Like the concept checks that are in the slideshows, like is there one
Oh,
for
yeah. It's coming. It's coming.
Oh.
Yeah. After I finish the whole thing, though. Yeah. But everybody okay with this so far? All right. Hope so. So let's see. Infecting bacteria with S35 labeled virus. Okay. So look at the S35, right? S35 means only the protein portion is labeled. Okay, and then the DNA is not labeled. Only the protein portion is gonna give off radiation. It's gonna glow, if you would. So if you use the S35 to infect the bacterium and then you spin it down afterwards, do you expect the radioactivity to be in the top part or the bottom part? Well, we know that it is the DNA that went into the bacteria, because now we know it's DNA that's the hereditary material, right? So it's not surprising that the black part, the DNA, went into the bacterium and it's going to be at the bottom. The protein shell that's on the virus is going to stay outside. So in their experiment, that's what they observe. Right? Radioactivity only detected in the top layer. Okay. Now, what this shows you, what this tells them actually, is that the protein didn't go in. Right? That's the only thing that they could conclude from here. The protein did not go in. This does not actually show that the DNA went in, right? Because I cannot see where the DNA is here, right? Without the radioactivity. Okay. This only shows that the protein did not go in. Okay, so conclusion. Therefore, protein did not go into the bacteria. Okay, but we don't know if the DNA went in, which is why we need to do the second part of the experiment, which is infecting bacteria with the other group, right? The P32 labeled virus. Okay, there's your test tube. There is your virus with the labeled DNA, with the radioactive DNA. And this time, where do you expect to find the radioactivity? Outside of the bacteria or inside the bacteria? What do you think? Give me something here, guys. Outside, inside? Yep, inside is right. Thank you. Because the DNA went in, right? So radioactivity only detected in the bottom layer. Therefore, DNA entered the bacteria. So when taken together, like take this result and this result, conclusion of their experiment. DNA is the hereditary material. Okay? They showed it. And then you have to have both results. You can't just make this conclusion based on one experiment or the other. You have to have both. Because over here, if they only had the results of this experiment of the one on the right side, yes, we saw that the DNA went in, but how do you know the protein didn't go in, right? But when you combine it with the result here, you can see this shows the protein stayed outside and only the DNA went in. Okay, so that was Hershey and Chase experiment. The purpose was to find out whether it was DNA or a protein that is responsible for hereditary material, and after doing the experiment, they concluded it was the DNA. That's the first experiment. We're gonna do 2 experiments today. Any question? Okay, well, there's a poll question somewhere here. Yeah, let's just skip to it and see. Let me launch the poll for you. Okay, what do you think? Okay, I only have 4 responses, unfortunately, and it has been a minute and a half already. This is not a difficult question. It's asking, why was radioactive sulfur used? Well, sulfur is unique to protein and not found in DNA, so these would not be the answer. And they were labeling the viruses, not the bacteria. So the correct answer is A. Okay. Hopefully that makes sense to you. If not, if you arrive late or missed the experiment, please look at them later. Okay, this is the bacteriophage that I was telling you about. You can see the protein and the DNA in separate parts, and then we already discussed that, so I'm not going to go through it. This shows you the experiment, and we also already wrote it down. So you can go through these diagrams yourself to see if it makes sense to you now. That's a picture of the actual virus. Okay, that's what they look like in real life under the microscope. Well, it's pretty cool. And you can see the DNA being injected into the bacteria. This yellow thing here, that's the bacteria. And then all these viruses are trying to inject their DNA into it and make more virus parts. Okay, and after they do that, the protein shells stay on the outside, right? Okay. So the next key thing for scientists to do now that they know DNA is the hereditary material is to find out the structure of DNA. So Rosalind Franklin was actually the scientist that had the data to first suggest that DNA is the double helix that we all know about today. Yeah, so using X-ray, it's called X-ray diffraction, which means she sent a beam of X-ray to the DNA sample, and they analyzed the crystal structure of a DNA. And based on that data, she was able to tell that the DNA is a double helix. But before she could finish her work, she actually died of cancer, probably because of exposure to the X-ray while she was, experimenting with it. Safety was not really very well regulated, I guess, back then. And so, she had too much exposure, and she couldn't finish her work. And then Watson and Craig are the ones that continued her work. Okay, and ultimately they built the first scale model of the DNA double helix, and you will find this picture in every textbook. And they won the Nobel Prize for the work. But a lot of people are saying, it should have been Rosalind Franklin because like she's the one who came up with the data, right? That first suggested it was the double helix. So that's the data from Franklin's experiment from the X-ray. So now they know it's double helix. Let's recap some of the things that you should already know for DNA. I'm just gonna go through this part quite quickly because you just had your test 2 weeks ago. Still should be pretty fresh in your mind. We know A pairs with T and G pairs with C, right? So we just call it the base pairing rule before, but actually it's called the Chargav's rule, named after Erwin Chargaff, the person who discovered this thing. And what's also interesting is that the DNA, not only are they parallel to each other, but they run in opposite direction, right? So we call that anti-parallel, right? Anti-parallel. Now, imagine driving on the highway, right? Like driving on the 404 or something, okay, or DVP, right? So we have one lane that goes from north to south, right? And then the other lane, which is parallel to it, is gonna go from south to north, right? Like the traffic is opposite, yet the two lanes are parallel to each other. So the northbound and the southbound, so to speak. And it's kind of the same thing over here. Instead of using north and south, when we refer to the DNA strands, we use numbers. Okay, so one strand we say 5' to 3'. That's what this little apostrophe is. We say prime. So one is 5' to 3', just like north to south on the highway, and then the other one is 3' to 5', right? South to north. Right? That's what these numbers are referring to. Okay? It's the directionality of the DNA. You don't have to know why it is 5 or 3, but you have to know one is 5 to 3, and then the other is 3 to 5. That's going to be important for some of the things that we're going to be talking about later on today. Okay, so it just gives a direction to each strand. basically. We did some of these calculations before. I'm not going to go through it again. You should already know this. We did this several times already. Base pairing rule allows the DNA to be complementary, which means if you know one side, one strand, you will be able to find out what the other side is. Okay, just because A always goes with T and C always goes with G's, and what's holding the bases together are the hydrogen bonds. Right? There are 2 hydrogen bonds between A and T, and 3 hydrogen bonds between C and G's. So it's much stronger, the forces holding them together, if there are more C's and G's. Right? These are just old information. Right? We talk about the anti-parallel already. Talk about the 5' and 3' already. And you can see here, that's the drawing. The backbone of the DNA is just made up of alternating phosphate and sugar, right? The sugar phosphate backbone. And then over here, we have the nitrogenous base. And so one strand is 5' to 3', and then the other is 3' to 5'. But the distance between the 2 strands are always the same. It's always consistent. Okay, and of course you twist it up to form the double helix. All right, next we have to talk about DNA replication. So here is a cell. We call this the parent cell. Now, some people get a little bit confused by this term. Parent cell is just the original cell in your body. Okay, it's not talking about your parent's cell. That's not what it means. This is just a cell in anybody's body. Okay, and this cell is going to basically undergo DNA replication, which means it will copy all of its genetic material. Okay, and then it will split into 2 genetically identical cells. These are called daughter cells. Daughter cells. Okay. So how many chromosomes do we have again, human cell? Anybody remember? That's a good number to remember from now on. 26. Okay, anyone else? 36. We're getting there. If we continue to trend, the next person will get it right. How many chromosomes do we have? 46, that's right. Okay, so we have 46 chromosomes, 23 from each parent. Okay, 23 from mom, 23 from dad. Together we get the 46 chromosome. Okay, so the daughter cells must also have 46 and 46, which means before you split the cell, you must duplicate your genetic material. Okay, you must copy all of it. Right? Otherwise, if you just start with 46 and you split, then you end up have 23, 23, right? And then like you would keep on reducing your genetic material, and you will be left with none in the end. Okay. So how exactly does a cell copy its genetic material? People didn't know. Okay. There were 3 models that were proposed, but people didn't know which one was correct until Messelson and Stahl did an experiment to show which model is correct. So let's take a look at the 3 models here. Oh, sorry, I went ahead of myself. This is the purpose of DNA replication. I already told you, right? We need to duplicate all the genetic material so that the daughter cells are going to be clones of the original, okay? Genetically identical. All right. So here are the 3 models of DNA replication. Okay, 3 models of DNA replication. We have the parent cell, okay? And in the semi-conservative model, what happens is you pull apart the DNA strand, and then you just use each half to build a new strand. Because remember, it's complementary. So if you know half, you can build the other half. So in the semi-conservative model, like half of the DNA is made up of the original material, and then half of it is made up of new material. Okay? If you do this again, so you separate the pink and then you separate the blue, the pink and the blue will build another blue strand, and then you will have a pink and a blue. So this is semi-conservative. Half of it is new material, half of it is old material. Conservative model is like this. You take the original and then you just keep the whole thing as is, but then you make a copy of it. Okay, so it's no longer half and half. It's either entirely original or entirely new. And then dispersive is similar to semi-conservative in that you have half new, half old, but then the new material is not restricted in a strand. Okay, you see how here in semi-conservative is either the whole strand is new or whole strand is old. But here it's mixed together. Right? Some are new, some are old. And I know that's really confusing to visualize, so I have this slide to help you. It took me half an hour to make this slide, okay? So I hopefully it will help you understand better. So here we have a picture, okay, the original copy. Right? So if I make a photocopy of it, okay, so I have the original, and then now I have the new copy. What do you think? Which model do you think this is? Is this That's the first one, okay? Is this the semi-conservative? Is it conservative, or is this dispersive? What do you think? The original, and then you photocopy like that, and then now you have two copies. Which model do you
Conservative?
think? Conservative is correct. That's right. Right? So this is conservative. Okay, that's what it would be like if we are doing this with actual paper, okay? Now this one is like this. You take the original and then you rip it in half, and then you stitch a copy of the missing half, and then now you get two copies. Okay? This would be semi-conservative, right? And then over here, this is like, you rip it up into tiny pieces and then you stitch them back together, patchwork, right? So it's a mix of new and old, but not exactly half and half for one side, right? Like it's new and old, like all over the place, right? So this would be dispersive. Okay. Now, in case you don't understand, you got lost in what we're talking about. These are the three proposed models for DNA replication. Okay. People didn't know which one is correct. Okay. And so they had to do an experiment, right, to find out. And that would be the Messelson and Stahl experiment, which is what I'm going to show you right now. Okay. For this to make sense, you need to have ideally three colors. Okay. Blue, red, and purple if you have, right? If not purple, then some other color. But you need to have three colors for this thing to make sense to you. Okay. All right. So Messelson, double S, I think. Um, nope, single S. Sorry. Messelson and Stahl experiment. Sorry, I keep on spelling their name wrong. Purpose. Can someone tell me what's the purpose of their experiment? After I went through all those explanations. Can you tell me why are they doing the experiment? What's the goal? What's the purpose?
To determine the proposed DNA replication, like which one's right.
That's right. Okay. To determine, thank you, the correct model of DNA replication. Is it conservative? Is it semi-conservative? Is it dispersive? What's going on? Let's find out. Okay. So how did they do it? So I'm just gonna write some point form notes for you. They grew E. coli in heavy nitrogen. Okay. Heavy nitrogen is N15. If you look at the periodic table, the regular nitrogen has a atomic mass of 14. That's the most common nitrogen, the regular nitrogen, if you would. The isotope, the heavy nitrogen is N15. Okay, so they grew E. coli in heavy nitrogen for many generations. Okay, like bacteria, when they divide, that's called one generation. They divide again, second generation. Every time they divide is a generation, right? So they grew it in N15 for many generations. Okay, this ensure all the DNA contains N15. Okay, so this is E. coli, and then all the DNA is now N15 DNA. And so what they did was they extracted DNA and then they have this test tube. Okay. The easiest way to explain this is the test tube has something kind of like corn syrup, okay, in it, and then you put the DNA in, and then depending if it's heavy or not, then it will be at different parts of the test tube. Okay, so N15 is really heavy, so you would have a band here. Okay, DNA, and we can say N15 DNA, right? Sinks to the bottom. Okay. If you have DNA that's lighter, it will go to the top. Otherwise, it's at the bottom, right? Any question about the setup so far? Again, please ask. Yeah, go ahead.
Is there a reason why they had to use the N15, or could they use something else?
Instead of, do you mean like why don't they use like phosphorus or something like that? Is that what
Yeah,
you
like
mean?
why did they have to use the nitrogen 15?
Yeah, I think the nitrogen is just more accessible at the time, easier to
Oh, okay,
prepare.
so it's just
Yeah, it's just
ease. Okay, it
convenient.
wasn't.
Exactly. No,
Okay.
yeah. All right. So what happened next? Right? Remember, the whole class of the E. coli has the N15 now. And so what they did next is they transfer the E. coli into regular nitrogen. You grow bacteria in a dish, right? So you can control what's in a dish. Like it either has like N15 or N regular N. So this regular nitrogen is N14. N14. And let them grow for one generation before extracting the DNA. Yeah. Okay, so this is generation 0. We'll call it generation 0. That's the beginning. That's the E. coli that have been growing in N15 for many generations already. Okay, so there you go. That's the DNA. So let's take a look at what the 3 model predicts. Okay. Let's say we do the conservative model. Conservative model. What would it look like? Well, conservative means I am gonna have the original, which is all just N15, right? That's the original one. And then the new one, the new DNA is gonna have to, get nitrogen from the environment to build that new DNA. And there are no more N15 available because they switch it to the N14, right? So the entire new DNA is gonna be N14. Do you understand? So if they extract the DNA at this point and then they put it on the column, where do you expect to find the DNA band? Top, middle, or bottom?
Like where the N14 would be?
Sure, where's the N14? Is that at the top, middle, or bottom?
Well, the N15 would be heavier, so that would be at the bottom. So the N14 would be near the top.
That's right. So there is going to be the N15 at the bottom and then the N14 at the top. If conservative model is correct, this is what you would expect to see. Okay? Let's take a look at the next model. What if it was semi-conservative? Right? Semi-conservative. Right? What would it look like? Well, semi-conservative means I am going to have one strand that's original, and then the other strand is going to be new, right? Okay. I'm not gonna draw the lines because it's too confusing. Okay. The point is, you will have in the same DNA molecule, you would have half of it being N14, the other half being N15. Okay. if you extract the DNA and you put it in the column, where do you expect to find the band? The top, the middle, or the bottom?
The middle?
The middle. What about the rest of you? Do you guys understand? Oh, thank you, Kiri. But everyone else, you guys with me? Like, give me some feedback here. Like, you know, are you guys all on the same page? You understand why it is in the middle? If not, you can tell me and I can explain it. Sure. Okay, let's backtrack a little bit. Do you understand why this one is at the top and the bottom? Okay. Before we talk about the middle, I just want to make sure that you understand that the lighter one is going to be at the top, right? Because it's less heavy. But then the heavy one is going to be at the bottom. All right, let's go back a little bit. I mean, if one of you is confused, probably several of you are confused. So they grew the E. coli in N15. So for the longest time, like you let them grow for I don't know 10 generations, okay? And all is available in the nutrient pool is the heavy nitrogen. So every time they grow, every time they replicate, they're gonna only pick up N15. So all their DNA has the heavy nitrogen. Okay, that's so like generation 0, everybody here is gonna have heavy nitrogen. Right? So when you put things into the test tube and you spin it down, basically, heavier things always goes to the bottom, okay, and lighter things flow to the top, basically. Right? And because N15 is the heaviest, that's why if you only have N15, it would sink to the bottom. Okay. So in the conservative model we saw earlier, that means when you copy the DNA, you will keep the entire original strand, okay? And then you just make a copy of it. The copy is made of entirely new material, and the original is made of entirely, well, old material, if you would. Since they transfer the E. coli into a new medium that's only containing N14, any new DNA that you make at this point can only consist of the N14. There are no more N15 available, right? Which is why you will have two kinds of DNA in your dish of E. coli, right? Old DNA, which is heavy, and then new DNA, which is lighter. So when you take the DNA of all the E. coli after one generation and you put it in a test tube, you will have two bands. The top band for the lighter DNA and then the bottom band for the heavier DNA. That's what the conservative model looks like. For semi-conservative, rather than taking the original and then you make a clone of it, right? Like you separate the two strands and then you build the other strand using new material. So in the end, your DNA is hybrid. It's half old, half new. So half of it is 15, half of it is 14, right? So how heavy is it going to be? Well, it's not going to be as heavy as the 15, and it's not going to be as light as the 14. It's going to be somewhere in between, right? Like 14.5. So that's why when you put it in column, you only have a single band in the middle, not the top, not the bottom, because the DNA is now hybrid. Okay, is that a little bit better? Does anybody have any follow-up question at all? Okay. Now the last one is dispersive, right? The dispersive is probably the hardest to draw, but it looks something like this, right? You would have um, the strand that's like a mixture of new and old, right? A mixture of new and old. They are not restricted to a to a particular sign. That doesn't even look too good. Here, let me try that one more time. I'm just gonna draw it out first, like this. Excuse me. And then I'm gonna add red like here and there. It's gonna be like all over the place, right? It's mixture. You have two of these things. Now, please help me out here. Let's do a poll. Okay. If I spin this down in the column, where would you expect to see the bands? If you think it's more than one answer, you can do that. Okay, but tell me, is it at the top, middle, or bottom? Okay, maybe a couple more seconds, and then I'm going to end the poll. So please lock in your answer. Well, I certainly had more people answering this time than earlier today. Well, the correct answer is the middle. Okay, the middle. Okay, we don't have DNA that's like just blue. If it's just blue, that's when it will show up at the bottom. If it's just red, it will show up at the top. But this is mixed, right? You got some new, some old. So again, it's not as heavy as 15. It's not as light as 14. It's going to be somewhere in the middle. Okay, so that's the situation here. All right. So here are the three possibilities. And then let me write out for you. Okay, so like this was not observed. Okay, they didn't see this. Okay, they did the experiment. They let the E. coli grow for one generation. They extracted the DNA. They spin it in the tube. They did not see this. Okay, which means conservative model is not correct. They saw a single band. They saw a single band, but that doesn't tell them whether it's the semi-conservative or the dispersive model. Okay, so let me write down for you. Only a single band was observed. Right. Therefore, conservative model was rejected. Okay. But they still don't know whether it's dispersive or semi-conservative. Okay. So what did they do that next? They repeat the experiment with another generation. Okay, so these are generation 1. Okay. Now you let them grow again. You let them grow again. You do a second generation. Okay. Now try to, oh, the program's gonna crash soon if, there we go. Okay, hold on. See if I can restart it. Okay, well, we didn't lose too much. So that's good. Okay, so they're gonna repeat the process, and this is what it would look like. So semi-conservative, right? I'm gonna separate the two strand. So I will have one strand that's blue, one strand that's red. Okay, let's focus on this one first. And then I'm gonna build a new strand. Okay, will the new strand be red or blue? In this case, just think about it in your head, okay? Do you expect the new strand to be red or blue? Remember, they have been transferred into the new medium, so only N14 is available, right? So like you would only be able to use N14 for the new strand. Okay. So here you would end up having one that's mixed and then one that's entirely new. Same thing for this one. We separate the blue and the red. And then we use the available N14 to build a new strand. And over here, if you take all these DNA and then you run it into the column, where would you see the band? Well, I would see a middle one, right? Because of this and this. This is the middle band if you must know. And then the one that's entirely red, that's going to be on the top, right? Okay, two bands. And 14, and then we have the 15 and the 14 in the middle. For dispersive is actually the same thing, right? Like this one, you would get two more, and then this one you would also get two more. Right? And then if you separate the DNA on the column, because they are still mixed together, it's still, not exactly all red, all blue, it's somewhere in the middle. That's why you're only going to see one band here as well. Okay. And then what did they observe after second generation? Okay, so this was not observed. Okay. Two bands were observed in the after the second generation. And so the correct model, conclusion, semi-conservative model is correct. All right. I know that was a lot of information. Go through it again when you have time. There's not going to be a lot of question on this thing on the test or quiz, but one or two maybe. So let's pause there. We will continue in 5 minutes. Okay, take a break. Okay, let's keep going with this. Here is a question that you might see. If DNA replication follows the conservative model, which of the following would be the expected result from Messelson and Stahl's experiment. Right? So why don't you take a moment and think about that? Take a look at the notes that you took and see. Okay, launch the poll. Anyone else? Right, I think I got my usual 8 participant. Um, that's the result you gave me. The correct answer is A. Correct answer is A. Okay, let's go back here for a second to see the semi-conservative. Oh, the color here is backwards. Maybe that's going to be confusing, but let's look at here. Right, this is the conservative model. If I let this go for another round, right, you would have the blue one. Right? The blue one, you would keep it exactly as it is, and then you make a red one. So now you have a blue and a red. And then here you would have the red, and then you will make another red, right? Because all there is available is red. So if you do this again, it will still look exactly like that for the second generation. It's not going to be in the middle. For the band to be in the middle, you have to have both red and blue in the same molecule. But that's not what semi-conservative model means, right? Semi-conservative model means I keep the thing exactly as it is, and then I just make a copy of it. Think of the teddy bear thing that I showed you, right? You have the original color copy, if you would, and then you make a copy which is just gray in color, right? So now if you do this one more time, think about it. How many copies? What kind of copies you're gonna have? You're gonna have the original color copy, and then you will have gray, gray, gray. Agree? Right? You're not gonna have a piece that's like half color, half gray, because that's not what the conservative model is like, right? So in the end, you will still have two bands. Okay, so that is the correct answer. Okay, you just go through it a couple more times yourself, see if it makes sense. If not, talk to me. I'll try to explain, um, to you again. Next, we're gonna move on to the last topic for today. We're gonna have to take another big note. That is called the mechanism of DNA replication in prokaryotes. Okay? Mechanism just means like methods, right? Like how does it happen? Essentially, that's what it means, right? DNA replication in prokaryotes. We are, of course, eukaryotes, but it's easier to understand the prokaryote first, and then we will extrapolate to see how it works in eukaryotes. What you're looking at here is DNA. Okay, and all these circles, these are called origin of replication. right? So you have to open up the DNA, and then you use each half to build a strand, right? We already figure out it's semi-conservative, so it's not surprising that you have to separate the two strands of DNA first, and then you use each half to build it. Okay, let me try to show you this on the note here. Okay. Let's see if this makes sense to you. Sorry, they changed the layout after they updated the program, so I don't really know where things are anymore. Okay, so this is the parent DNA, two strands. Okay, to make it easier, I'm not drawing the double helix. Okay, I'm rounding it. All right, so it looks like this. This is the parent DNA. And what we need to do basically is we need to create some bubbles. Okay, we're going to create some bubbles. There's one bubble. There's another bubble. Okay. These are called, again, origin of replication. That's where the replication starts. Okay. It creates, sometimes it's called like a replication bubble. Okay. There is another origin of replication. And then once you open it up, you will be able to build a new DNA strand, right? Build a new DNA strand. Like if you think about like a zipper, like your winter jacket, right? Okay, winter jacket. Like this happens to my kid's jacket all the time. Like sometimes, like the zipper thing, okay, like you zip up the jacket, and then it just kind of pop open in the middle of nowhere. Okay, like the zipper just unzip out of nowhere in the middle of it, right? And then you can stick your finger in and then just run your entire finger up and down, and then it basically unzips the jacket. I don't know if you can visualize what I'm saying here. Okay, but that's basically what happens. There is a replication bubble. It pops open, and then you can just expand the bubble, right, by running your fingers through it in both ways. And then eventually what happens is like the bubble gets bigger and bigger. Try it one more time. Right, as you build a new strand. Right? It just keeps on expanding. And then before you know it, you would have two strands. Okay, so these are daughter DNA. Okay, that's basically what the semi-conservative model is. Now I can do this with you again with actual nucleotides. So if this is one strand, we can write down what's on the other strand. Okay, you don't have to copy this down, like you can just watch. So this is what happens. Look, during DNA replication, you will separate the two strands, separate them, and then you will build the missing half. Your body knows A goes with T, T goes with A, and so on and so forth. Same thing over here. You're going to build the other half. Right? So you end up having 2 strands. This is semi-conservative. Questions? No question. Okay. Now I already showed you this, right? The DNA parent molecule unwinds. The 2 new daughter strands are built based on base pairing rule. It's super fast and super accurate. In prokaryote, we're talking about 1,000 base pair per second. Yeah? And then the whole DNA in E. coli, for example, it's about 4 million base pair. Okay, so it's like typing 1,000 words per second and not making a mistake, right? Medicine said, what if they mess up? Yep, they do mess up. But as we will see, they have spell check mechanism. That's actually what they're called. It's like when you're typing on your computer and then you made a mistake, like the Microsoft or whatever will highlight it for you, right? And then you will go back and then press backspace and then make the change, right? So they actually has that capability of auto correcting. Now, does the auto correct always work? It works reasonably well, okay, but it's not perfect, right? Sometimes they are not able to catch the mistake, and that's how people get a mutation, a permanent change in their DNA because when they copied it, they made a mistake. Now, the mistake doesn't have to happen during DNA replication, as we'll learn later. It could happen for other reasons, but that's one of the sources of mutation. Right? You basically
Question.
copied it wrong. Yeah.
So like one day the mistake can just happen and then you're changed forever, or
Yeah, so like we're gonna learn more about it later. But if you have a mole somewhere in your body, that's essentially what a mole is. Okay, like when your
uh,
skin cell replicate, at some point during the replication, they made a mistake, right? The DNA changed, and then it causes the pigment to become different, and then so like this is the original cell, and then like you divide. Okay, you get like another cell here, and then you divide, and then you get cells. So these are all the same, but then maybe there is a mutation here, right? They didn't correct the DNA, and then this becomes like one cell that's like different pigment now. And then every cell going on from there is gonna be like a black cell, right, so to speak. And then that's how you get a mole, right? It started with one change, and then every descendant from that one mistake is gonna look like that now. And then that's essentially what a mole is. How do DNA change for someone to get Down syndrome? Yeah, we're gonna learn about that. That is not mutation. That is when the usually the mother when the female makes the egg, because in sperm and egg we have to reduce the chromosome number by half. Sperm has 23, egg has 23, 23, 23 gives 46 to the baby, right? But if chromosome fails to separate, then you might end up having extra chromosome in some cases. So for Down syndrome, it's an extra chromosome 21. We will learn that later on in lecture 8. Okay, so do stay in the course. A lot of exciting things coming our way in terms of other concepts that we have to learn. Okay. I went on a tangent here, but let's come back here. The copying is, like I said, is really, really fast. 1,000 base per second. We're talking about 4 million bases in total for something like E. coli. For humans, is slower. Okay, we are go around like 40 bases per second. I mean, it's still pretty fast, but it's certainly not as fast as the bacteria. And we actually have a lot more genetic material. Okay, 4 million base pair in E. coli. How many base pair do you think in human genome, in human DNA? What do you think? Like ballpark, what number? Just throw it out there if you want to guess. You guys up for billions, right? 3 billion. That's the approximation, okay, of how many DNA, how many ACTGs you have to copy. Okay, we have 3 billion of these things. So how do we make this happen? Well, we actually need to have a bunch of enzymes and a bunch of proteins to make this work. Okay. I already showed you about the origin of replication, where you form the bubbles and then the bubble expands laterally, and you end up, fusing the bubbles and getting two strands. Here are a bunch of enzymes that are involved in DNA replication, and we are going to take a note and introduce all these to you. I'm not sure if I can finish the note. It's there are only like 20 minutes left. We'll do as much as we can. Whatever we don't finish, we'll finish on Thursday. Medicine says someone counted. Yes, you actually I will show you guys in lecture 9. The human genetic material is available online. You can go online. I'll show you later on. And you can start on the first nucleotide of chromosome 1, and it's all listed out. Like chromosome 1 is A, C, T, T, G, C, T, 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 all the way to the end of chromosome 1. And then we know exactly the whole genome of all the human chromosomes. It's really cool. And we need to know that in order to do genetic engineering, right? To do gene therapy, for example, like you need to know which gene goes where and how to modify it, right? It's really exciting stuff. I get, really pumped up talking about these things, but we'll get to it later. Right now, we have to do the DNA replication. Okay, DNA replication in prokaryotes. Okay, it's similar in eukaryotes. It's just much more complicated. So we're gonna start with the prokaryotes. Please take out a new piece of paper. Okay. I'm gonna draw here. Your diagram should be about this big. Okay, like it should look something like this when you draw it. All right. So let's just do a small version here first. Okay, this is the DNA, and this is again the origin of replication. Now, earlier today, I was telling you that DNA has directionality to it, like northbound, southbound, but we use 5' and 3'. So if this is 5' to 3', then the bottom has to be 3' to 5', right? Now, don't get me wrong. It's not always 5 and 3 on the top. Like these are just random, right? Like I mean, if you flip the DNA, then it's the other way around. So like, don't get too hung up on, whether it's the top one or the bottom one. The key thing is you have to know that they are like, opposite. So what we're gonna do is I'm gonna focus on this part. Okay, I'm gonna focus, zoom in on it, and then we're gonna get this thing. Okay. Like that. Sorry. Like this. And remember, this is like the double helix, right? Okay, which I'm not gonna draw because it's confusing. Okay, but just know that it's like that. So this is 5'. This is 3'. 3' and 5'. Let me show you what I did with the other class. So like you can plan for space. Okay, like where is it? Like we have a lot of stuff to put on it. Okay, so don't do a tiny diagram. Okay, make it big. Like I said, like this should take up like your entire page almost. Okay. Now I'm just gonna put some alphabets here to help us, with some nucleotides to help us see what's going on. Um, it's completely random, okay? But on the other side, it cannot be random. It has to be complementary, right? Um, please double check my work here. Make sure that I didn't, made a mistake. Okay, I think I did it okay. Like it's just base pairing, right? You see A pairs with T, T pairs with A. I'm gonna give you a moment to copy all that. Like obviously there are more nucleotides, right? Like go all the way to the end, right? But to keep it simple, I didn't draw them. And also we're just looking at half of the bubble here, right? This like it's gonna go on like that, right? But again, I'm not gonna draw it because it's confusing. It's already a lot of information. Okay. So this thing right here, this is called the replication fork. Because I mean, like it totally looks like a fork, obviously, right? So the bubble has two forks, one on each side. All right. So how do we do this? The first enzyme that we need, and I hope you have color for this. Okay, this is like a lot of stuff to draw. There has to be an enzyme here. Okay, so this enzyme is called helicase. Helicase. Okay, it is an enzyme that is going to unwinds the DNA. Okay, and it will continue to unwind the DNA after it creates the bubble. Think of it as the thing that you zip and up and down your zipper. Okay, the thing that you're holding on. I don't know what you call that thing. Okay, that's the helicase. Okay, you can unzip the zipper. You can unzip the DNA with the helicase. Okay, so like on the other side, you're gonna have another helicase, right? That's zipping the other in the other fork. That's it. That's helicase. Okay. Now further down the DNA, you're gonna have another enzyme here. Okay, so this other enzyme, uh, you can use whatever shape you want. It doesn't really matter. Okay, this enzyme is called what is it called? Gyrase. And the function of gyrase is to relieve tension that builds up in DNA as it is unwinding. Okay. Don't draw this, but pause for a second and take a look at what I have to say. Um, this is the bubble, right? The bubble. And then remember the rest of the DNA is still like all twisted up, right? All twisted up. So I want you to visualize this. Think of this as like a rubber band, okay, that you're holding on to one end, and then over here, if you pull these in opposite direction, right? Okay, slowly pull it in opposite direction. and this is the rubber band, what's gonna happen? Well, as you unzip more and more, like the rubber band is gonna twist up and form these knots, right? Can you visualize that? I mean, I don't have a rubber band here, but like try it. Like take a rubber band after class. You cut the rubber band once. Okay, so now it's open. You tell someone else to hold this end, and then you hold on to the other two ends here. And if you keep on twisting it, it's just gonna get like all tangled up and tied up in a knot, right? And that's essentially what's happening with your DNA here. Okay. The DNA, as it unzips, the rest of it has a lot of tension building up in it. And if you don't relieve those tension, eventually the DNA will break off, will snap off. Okay. And that's a bad thing. Okay? We don't want DNA to break. That's why we have to have the gyrase. Without the gyrase, your DNA is likely to break. All right. I think I have a picture of that in the slide, like here, right? Like you see the rubber band is like all twisted up, right? Yeah, so we don't want that to happen. That's why the gyrase will relieves is that how it's relieves any tension from unwinding of the DNA. Okay, I hope you are with me so far. Now, the two strands of DNA excuse me, the two strands of DNA are actually attracted to each other, right? Because of the hydrogen bond. So, the A and the T's wants to go back together and the C and the G wants to go back together. But we cannot have them go back together. We need them to be separated so we can gain access to the nucleotides so we can build a new strand. That's why we need to have proteins that would keep them apart. Right? So like there are these little proteins here. Okay, you can draw some on the other side as well. Okay, these are called single strand binding protein. Okay, and their job is to prevent the DNA strands from coming back together. We don't want them to go back together. We want them to stay separated so we can build a new strand. So those are the three key things that are happening at the beginning, huh? The gyrase, the helicase unzipping, the gyrase relieving the tension, and then we have the single strand binding protein keeping the strands apart. All right. So we'll leave this picture for a little bit, and I'm gonna tell you about something else for the next little bit, and then we'll come back to this later. We have another enzyme here. Let's, I don't know what other color. Let's use like green here. Okay. You know what? Let's just write it in black for now. There's an enzyme called DNA polymerase 3. Okay. If you're wondering where is number 1 and 2, don't worry, they're coming. Okay, but let's do DNA polymerase 3 first. This is the enzyme responsible for building the new DNA strand. ASE is enzyme. So this is an enzyme that builds DNA polymer, right? That's essentially what it means, DNA polymerase. It's an enzyme that creates the DNA polymer, the nucleotide, essentially. Two limitations. Number one, cannot initiate the process, right? Cannot start, cannot initiate. Right? It doesn't know where to start. It knows how to build it, but it doesn't know where to start. That's a problem. And number two, can only build new DNA in 5' to 3' direction. Why DNA needs to has bubble? Because look, this is the original DNA, right? And because of the semi-conservative model, we have to use each half as a template to build it, right? So I need to gain access to the nucleotides before I can, build a new strand. That's why you need to make the bubble. By creating the bubble, now the polymerase tree can see the individual nucleotide, and it's like, okay, I see an A, I should put a T here. I see a G, I should put a C here. That's why we need to create a bubble. Does that answer your question?
Thank you.
Okay, great. Thank you. All right, so we have two problems here, right? Cannot start and can only build in one direction on the new strand. All right, let's try to fix some of these problems. DNA polymerase reminds me of my kids when they have to do their homework, okay? Like they know how to do the homework most of the time, but then like they wouldn't start doing it, right? They come back from school, took a shower and then come down. They're not doing their homework on their own, right? They're like they don't know where to start or something, right? So I have to sit down with them. I'll do 1 or 2 questions with them together, and then they could go on and finish it themselves. That's basically what's happening here. DNA polymerase 3 doesn't know where to start. Some other enzyme has to come and show them where to start and how to do it. Okay, so that would be the job of yet another enzyme. Okay, so there is going to be an enzyme called, hold on, I got to change the, how do I do this? It used to be so much easier. Oh, there it is. There's the wheel. Okay, so there is another enzyme here called, and again, you can use whatever shape you like. This is called an RNA primase. Okay, RNA primase, the job is to help start the DNA replication process for DNA polymerase 3. Okay, that's like me helping my kids with their homework. You know what? I'll do a few questions for you, okay? So it's like there's a G, so G goes with C, okay? And then there's an A, and what does A go with? Now I know what you're thinking. You're like A goes with T, right? Okay, which is fine in the case of DNA, but remember this is RNA that we're talking about, so there's no T. They will put a U instead. Okay, A goes with U, and then T goes with A, and then that's it. They're just gonna do a few. All right, I'm not gonna do the whole homework for them. They're just gonna do a few, and then DNA polymerase 3 will continue on. So this little bit here, this little bit that we just the RNA primase just created, that's called an RNA primer. It basically helps position the DNA polymerase 3. Do you understand? I feel like the program is going to crash soon again. Oh my goodness, super slow here. I'm not sure why. Hopefully it will hang on for another 5 minutes. Okay, so then we're going to have the polymerase 3 right here. That's the DNA polymerase 3. And then it's just going to continue. Okay, C goes with G, G goes with C, T goes with A, and then it will just keep on building and building and building into the fork. Okay, and of course, if you are keeping track with the direction, the original is 5 to 3 like that. So that means on this new one, what number should I? Oh, it crashed. Sorry. What number is it going to show up on this end? Hold on, let me just close it down first and see if it's going to come back. Oh boy. Okay, my question was, what number is going to show up here on this end? Please tell me. Is it a 5 or a 3 on this end? Hopefully, you are thinking of a 5. So that's 5 prime to 3 prime because it must be anti-parallel, right? Opposite to each other. Okay. And also, if you look at the limitation, the second limitation is it can only build new DNA in 5 prime to 3 prime direction. Okay. So over here, it's going from 5 prime to 3 prime. It cannot go backwards, in other words. Right? So what's going to happen on the other side? Well, on the other side, same thing. You're going to have another DNA primase. I'm not going to draw it, but I'm going to draw the primer here. Okay, so it's going to go like UAAG maybe. And then you will have a polymerase 3 that will continue to build this way. So what happens to all these things? How are we going to copy all these DNA as we open more and more of the fork? Well, you're going to need to have another primer here, like a GCC, right? Like that. And then the polymerase 3 will build this way. Okay. So as you open more and more, you're going to need another primer and another primer. But that's not going to be a problem for the top strand because the top strand will just keep on going into the fork, right? Do you understand that? So let me make a note of that for you. Okay. So this top strand, this strand here, this is called the leading strand. Leading strand. Because it is synthesized, it's built continuously. Okay. And it goes into the fork. It goes right into the fork like that. And the other strand, the one that's at the bottom, right here, this strand is called a lagging strand. And that is built in fragments. Okay. And it goes away from the fork. Okay, away from the fork. And just one last thing. Okay, These fragments, these fragments, they have a name. The fragments are called, it's named after the person who discovers it, Okazaki fragment. Now let's not forget at the bottom, the numbers are the directions, or the opposite to what you have at the top. And that is where we're gonna end today. This is not done yet, this story. We're gonna have to finish it up next time. You might want to reorganize this in your own way, and we'll draw more when we come back next next class. Okay. All right. Thank you. Have a good day, and I will see you on Thursday. I'll stay behind for a couple more minutes in case you have questions, but otherwise, have a good one.